Formation of the central nervous system begins very soon after fertilization. Somewhere about week 3 is when the central nervous system begins to develop. So it really depends a great deal on the steps and the processes that happen right after fertilization. So that's where we're going to start in order to establish a foundation for what's going on, and so that you can comprehend the various steps that go on during development of the central nervous system. We actually need to take a step back and look at the steps that happen right after fertilization. So that's where we're going to start. What you're seeing here is a colorized scanning electron micrograph of sperm swarming an egg, and this of course is going to lead to fertilization. Eventually one of these sperm is going to penetrate the egg, and that will be the cell that contributes its DNA to the fertilized egg, and the resulting offspring will have a combination of the mother's DNA, which is already in the egg, and the DNA that's in the nucleus of the sperm from the father. Fertilization typically happens in the uterine tube, and during the first stages of embryonic development, that embryo, now after fertilization it's a zygote and then an embryo, is going to be traveling down the uterine tube. During these early stages, as that zygote, the fertilized egg now becoming a zygote, is traveling down the uterine tube, it begins to divide. It starts off as a single cell, and the cell and the cytoplasm and all of the cytoplasmic organelles are contributed by the mother in the egg, and that cell begins to divide, and it undergoes a period of very, very rapid cell division. So rapid, in fact, that there isn't any time for those cells to grow. Eventually, they'll start to grow and divide, but at this point, the cell is producing all of the new cells that are going to be required to produce the new organism. These newly produced cells are called blastomeres. And again, since there's no time for those cells to grow in between division, they just start dividing and then they produce new cells, and those cells start dividing and produce new cells, and so on and so on. The cells begin to get smaller and smaller and smaller and smaller and smaller. And what you get eventually is just a ball of cells. You start with 1, it divides to form 2, those 2 divide to form 4, which divide to form 8, and so on and so on and so on. After about 72 hours, the zygote reaches a stage that we call a morula. And the morula stage is basically a cluster of cells. It begins at about 12 to 16 cells, which is what you're seeing here, a microscopic image of a morula, early morula stage. And as those cells continue to divide, at about the 4th day, the morula, late morula, now probably somewhere around 60 cells, enters the uterus. So what you have is essentially a ball of very small cells entering the uterus, and the next step is the development of a cavity within that ball of cells. So what you're seeing here, once again, is that initial diagram, and it's showing you the development of that morula, and eventually it gets to the point where you just have this huge ball of cells, and eventually you get a hollow space forming within that ball of cells. That hollow space is called a blastocele, and the name for the zygote at the stage of development is called the blastocyst, named for the presence of that blastocele. Very soon, as those cells keep dividing, you start to be able to see 2 distinct cell types visible within the developing embryo. What you have is this layer of cells around the outside. That was that layer that was initially surrounding the internal cavity. We call this population of cells the trophoblast. And then you have this cluster of cells that begins to develop within that space, and you develop this cluster of cells on the inside. And that cluster of cells on the inside of the trophoblast is referred to as the inner cell mass. And it's that inner cell mass that's eventually going to form the embryo. This stage lasts for about 3 days. So At about the end of the first week, beginning of the second week, is when this developing embryo is going to actually implant into the uterus. On or about day 7 is when you start getting implantation of this embryo within the uterus. And one of the first things to happen at about the stage of development is that inner cell mass starts to differentiate further. Within that inner cell mass, we can once again identify different populations of cells. We have a structure in the upper portion of that inner cell mass, and that upper portion is referred to as the epiblast. And below that, you have a layer of cells that is referred to as the hypoblast, and that hypoblast is going to go on and eventually form the yolk sac. And again, as we mentioned, meanwhile, as all of this is going on, the embryo is embedding in the uterine lining. So if we look at a close-up of an artist's rendition of the embryo at the stage of development, the cells of the epiblast and the hypoblast keep dividing, and eventually they form clusters of cells, and just like we saw with the initial embryo, eventually you start getting spaces developing within the epiblast and within the hypoblast. Now, the layers of cells that are directly in contact with one another are still referred to as the epiblast and the hypoblast, but once again, within these cell populations, you get these fluid-filled spaces, and eventually the upper cells, that space is going to form the amniotic sac, and as we mentioned earlier, the space within the lower cells is going to form the yolk sac. So essentially what you have are these two spherical, hollow balls of cells that are in contact with one another, and the point at which they are in contact is referred to as the embryonic disc. And it's going to be the two layers, the epiblast and the hypoblast, that are in contact with one another at the embryonic disc, which eventually form the embryo. So this was all going on during the second week of development, and you have other developmental processes going on external to the developing embryo. You start getting development of the placenta as a combination of maternal and embryonic cells, but we're not really going to concern ourselves with that because we're more concerned with the development of the embryo, which is of course going to lead into our discussion of the development of the central nervous system. So during week 3, again, is when a lot of the really important stuff starts to happen with regards to the development of the central nervous system. And I have listed a lot of the important processes that lead into this. The primitive streak leads into development of the primary germ layers, and then you have development of a notochord, and then neurulation and differentiation. And we're going to explain what all those mean in the coming slides. So back to our diagram showing the embryonic disc, and what we can see is the upper layer of cells, the epiblast, the lower layer of cells, the hypoblast, and on the right here you can see a cross-section of those. So the upper layer of cells here, the blue is the epiblast, the bottom is the hypoblast, and we're going to be discussing actions that go on with regards to those cells. The first thing that happens is formation of a structure called the primitive streak. And again, this typically happens in that second week leading into the third week of development. And what you get is a groove that develops within the cells of the epiblast. And it begins at what is going to eventually become the tail end of the organism, and it progresses towards the end that's going to become the head end. So at this point we have the first indications that we're going to get a organism that has a distinct head and tail end. And what happens is cells of the epiblast begin to migrate downward. So you get division of these cells, and these cells begin to migrate downward through this groove that we refer to as the primitive streak. And those cells are going to take up residence between the layers of the epiblast and the hypoblast. So what we're doing is we are producing a third population of cells. We have the epiblast cells, we have the hypoblast cells, and then between the two we're getting this third layer of cells migrating and eventually taking up residence between the two. These three layers of cells are eventually going to produce our three germinal layers. And those three germinal layers are the endoderm, the mesoderm, and the ectoderm. All of the subsequent tissues of the body are going to arise from these three germinal layers. So even at week 2, week 3 of development, we begin to have some very definitive differentiation of cells happening. The migrating cells eventually replace the hypoblast underneath the developing embryo, forming the endoderm. Then you get a middle layer forming in between, that forms the mesoderm. And finally, the cells that stay on top, that were originally the epiblast cells, become the ectoderm. So if we take another look at what's going on outside the developing embryo, we can see that there's been further differentiation of the placenta. And once again, we can see those three germinal layers of the embryo in the center. So back to what's going on within the embryo. You have that primitive streak that begins to run from the eventual tail end of the organism to the eventual head end of the organism. And as those cells migrate downward, once again, you get development of those germinal layers. And this is what's going to define the axis of the body. And there's a little lump at the front of the primitive streak that we refer to as the primitive node. And of course, that's eventually going to end up at the head end of the organism. After development of the primitive streak, you have development of the notochord from that mesodermal tissue underneath. But what's going to happen is you're going to get further differentiation of that ectodermal tissue above the notochord. And this is what is going to lead to our central nervous system. The cells in the middle begin to grow and elongate, and they form a thick region called the neural plate. And then there's a depression that forms in the center of that neural plate, and it forms a groove. And that groove runs along the same line as we saw the primitive streak. And this depression, what's eventually going to happen is the edges of those grooves are going to start pinching in, and eventually that neural groove is going to close. And what's going to happen is it's going to form an actual tube. And this neural tube is what's going to form the brain and the spinal cord, the components of the central nervous system. So here are the next stages of development. Again, we can see the groove getting deeper and beginning to close. And eventually, after about 20 to 22 days, you end up having that neural tube close completely. And once again, that neural tube is going to form the brain and spinal cord. This is the process of neurulation. Closing of this neural tube is very important, because if that neural tube doesn't close completely, this can cause very, very severe abnormal conditions like spina bifida or abnormal development of the brain, depending on where the point of the tube that remains open happens. So what we can see here is a few diagrams showing you the progressive development of the brain and the spinal cord from the neural tube. If we look at this left portion of the lower diagram, we can see just the basic neural tube. It's a hollow tube, and you've got the anterior end, which is going to form the head, and the posterior end, which is going to become the tail end. The next series of developments happen at the anterior end of the neural tube. You start getting enlargements. So the anterior end of that neural tube develops these enlargements. And you start with three initial enlargements. The anterior-most vesicle forms the first enlargement, called the prosencephalon. The second forms the second enlargement, called the mesencephalon. And the third forms the last of the three portions of the developing embryonic brain, called the rhombencephalon. So these are the first three portions of the brain to develop. Now, in a lot of cases, these three primordial regions are going to develop further into other portions. For instance, if we look at the prosencephalon, eventually that prosencephalon is going to form secondary developmental areas called the telencephalon and the diencephalon. The telencephalon is eventually going to form the cerebrum of the developing brain, and the diencephalon will form what eventually remains referred to as the diencephalon, the region that contains the thalamus, the hypothalamus, and the epithalamus. The mesencephalon continues to be referred to as the mesencephalon, and eventually that forms the midbrain. And finally, the rhombencephalon, the last part, the hindbrain, forms two more structures, the metencephalon and the myelencephalon. And the metencephalon and the myelencephalon are going to form the pons and the medulla, which are portions of the brainstem, and you will also get development of the cerebellum out of the metencephalon. So what we can see here are the first three primordial vesicles to form, and then further development eventually forms the regions that are going to form the adult portions of the developing brain. The fluid-filled spaces within those regions are going to form the ventricles within the brain. The lateral ventricles form within the telencephalon, the third ventricle forms within the diencephalon, the cerebral aqueduct within the mesencephalon, the fourth ventricle within the regions of the met and myelencephalon, and then finally below that you have just the spinal cord, which encloses the central canal. If we look at the upper portion, the upper diagram here, we can see that in addition to these vesicles forming, these enlargements, we can see that the anterior end of the spinal cord, the central nervous system, is also sort of folding. It's taking on the folding that will eventually produce the adult positions of the different regions of the brain. So if we look at this diagram, we can see that the original folding and flexures within that neural tube are going to line up the regions of the developing brain and position them so that as they grow and develop, they will develop in the appropriate places. The developing telencephalon is going to grow and enclose and develop on top of the developing diencephalon, and then the cerebrum, the pons, and the medulla are going to sort of scrunch up on one another, and as the brain continues to grow, you end up with the appropriate portions of the brain developing in the appropriate regions.